Hello everyone, welcome to Quantum Learning Academy. In this video we'll talk about the bioinformatics databases, and this is the second video and about the bootcamp that we have started, and in this particular video we'll explore about the bioinformatics databases. Since I mentioned in my previous video that the first segment of this particular bootcamp is about the bioinformatics databases, and we all know that it is very important for a bioinformatics student to actually explore and to actually know and to learn about how you can retrieve information and how you can extract information from these particular bioinformatics databases. Well, first we'll talk about the learning objectives. So this particular slide explains what are the learning objectives associated with this particular tutorial. So we'll first talk about what is actually a bioinformatics database, what are the core features of bioinformatics databases, and we'll talk about the importance of bioinformatics databases in biological research, and also we'll talk about the importance and the types of bioinformatics databases. Connected to the type of bioinformatics databases, I will practically demonstrate some of the examples of bioinformatics databases and how these bioinformatics databases are used. In the last part of this particular tutorial, we'll talk about the challenges and limitations of bioinformatics databases and the future direction in bioinformatics databases and its usage. So it's pretty much everything, and so let's get straight into the learning part. Now, the first question that comes in the mind of a student is, what is actually a bioinformatics database? Well, bioinformatics database is a collection of biological data that has been organized and formatted in a way that facilitates the storage, number one. Number two is the retrieval, and number three is the analysis of this information using the computational tools. And the reason why there are so many bioinformatics databases is actually these informations are derived from the high throughput experimental technologies such as DNA sequencing, gene expression profiling, protein structure determination, and prediction. So this particular slide explains the reason why there are so many types of bioinformatics databases and what is actually a bioinformatics database. And a very simple example, and I know every student, when we talk about the bioinformatics database, the NCBI pops out in their mind that, yeah, I've explored this particular database. So NCBI is a perfect example of a primary and a secondary database. Now, what are the core features of bioinformatics database? This particular slide help you to understand what are some of the characteristics and the identical features of a bioinformatics databases. And when you know about these particular features, you will not afraid to explore any type of database that you have confront in your future. Now, the first thing is searchability. There will be an option related to search. You can search. There will be a navigation bar. There will be some sort of interface that will help you to search in a particular database. The second point is the accessibility. You will have access to a lot of the information and in a particular database. The third thing is the data standardization. This point explains actually the format of the data that is stored in a particular database. The data will be in the standard format, and you will not need to actually go around to find a particular information. There will be a standard format, and you have to look for that particular format to know about that particular database. The second thing is the data visualization. In some of the databases of bioinformatics, and I think in every database there will be a section to visualize some information. And if we talk about the PDB, which is a protein database, and in this particular database you will have information about the 3D model of a protein. And if you talk about the NCBI, you will have information about the sequence, the nucleotide sequence, the protein sequence. You will have information about the messenger RNA, and etc. The fourth and the fifth and the second last feature of bioinformatic database is the data integration. You will have the option to integrate something. You will have option to input something and to get some output. If we talk about the BLAST option in the NCBI, where you need to put a particular nucleotide or protein sequence, and at the end you have to blast it against a particular database, and at the end you are getting a pairwise alignment between the two sequences, between the query sequence and the sequence that match with that particular query sequence from the database that you have selected. The last core feature of bioinformatic database is the annotation. Well, annotation means there will be a proper naming associated with a particular information. If you are talking about the DNA sequence, if you want to retrieve a DNA sequence, there will be information about the ORFs, there will be information about the exon, there will be information about the introns. So these core features actually help you to understand what sort of information and what kind of information and in what design the information will be there in a bioinformatics database. Now, what is actually the importance of bioinformatic databases in biological research? So here are some of the key ways bioinformatic databases they play a very vital role in biological research. When we talk about the data organization and standardization, so these bioinformatic databases, the data is organized and it is in the standard format, so it helps you to retrieve a particular information in a very efficient way. The second point is the facilitation in the data analysis. Like if you want to analyze something, you can do that using bioinformatic database. And just to give you an example, if you want to do a network analysis, you can do that using the cytoscape. It's a particular database which are utilized when it comes to the network analysis. Now, in the generation of new hypotheses, like just to give you an example, if you have identified a particular gene which is unknown to you, you can utilize different kind of the databases like expression atlas, like GPL2, to know about the role of these unknown genes that you have identified. So it helps you to generate a new hypothesis. Now, when it comes to the acceleration of new discovery, like if you explore a NASA GeneLab database, there are many experimental data which has been uploaded on this database, and you can actually download these data, and you can run and you can analyze it without any experimental work. You can just do the computational work, and you can come up with a new discovery. So these databases actually accelerate the process of new discovery. And the last point, in fact, it is about the promotion of data sharing and collaboration. It is important, and the essence of these bioinformatics databases is to share the data with the scientific community and to collaborate on the research perspective. Now, let's talk about the types of bioinformatic databases. There are many types of bioinformatic databases. When we talk about the type of databases on the basis of the information, like primary database, secondary databases, there are databases which are organism specific. There are databases which are disease specific. But here, this particular slide explain the databases on the basis of some features. Like if we talk about sequence databases, these are those databases that store the sequence, like DNA sequence, like RNA sequence, like microRNA sequence, like protein sequence. So the first type is the sequence database. The next type is the literature databases. Those databases that actually help you to retrieve literature about a particular problem or a particular hypothesis that you have come up with. Gene expression databases, these are very popular databases. If you want to know about the expression profiling of a particular gene, you can utilize these databases like gene expression databases. Variation databases, if you want to know about the the mutation in a particular gene. If you want to know about the indels, if you want to know about the single nucleotide polymorphism, you can utilize these variation databases to actually know about the reason behind the mutation. Ontology databases, the cancer databases, in other ontology databases are those databases that actually have information about the cancer. Like if you are working on a particular gene and it has a role in cancer, you can utilize these ontology databases to know about the genes that are involved, to know about the pathways that are involved, to know about the tumor progression, to know about how they are involved. So the ontology databases, they are very common when it comes to cancer biology study. The last and it's very important, the pathway databases. If you want to know about the pathways that are associated with a particular gene, that are associated with a particular protein, you can utilize these pathway databases to know about these all things. So this particular slide explains the types of the bioinformatics databases, and in the coming videos we will explore these types of databases in detail. Now, some of the examples of the bioinformatics databases, of course, we know about the NCBI. NCBI is a primary database, a big database, and we have secondary databases associated with NCBI like GenBank. If you want to know about the sequence of a protein, you can utilize the UniProt database. If you want to know about the 3D structure, the experimental 3D structure of a protein, you can utilize the protein databank. And if you want to know about the pathways that are associated with the genes and the genomes and the chromosome, you can utilize the KIG database. And if you want to know about the cancer pathways, the cancer related information related to a particular protein and a gene, you can utilize the gene ontology database. And if you want to know about the Mendelian inheritance in a main, if there is a particular information you want to explore about the Mendelian inheritance, you can utilize the OMIM database, which is a part of the NCBI. So these are some of the examples of bioinformatics databases, and at the end of this video I will show you some of the databases, and we will talk about these databases. Now, how and why we are actually studying the bioinformatics databases? Well, when it comes to the sequence analysis, these databases can help you to understand the sequence, can help you to predict a particular sequence, can help you to evaluate a particular 3D model, and it help you to retrieve a particular sequence. So these databases are very important when it comes to sequence analysis. Phylogenetic analysis, if you want to know about the lineage, if you want to know about how your sequence and how this protein is related to another species, how it is related to another lineage, you can utilize these databases, phylogenetic analysis. And if you want to know about the pathways that are related with your gene that you are working on, so these databases are helping in the functional annotation part. And pathway analysis we talked about in the previous slide. And literature mining, if you want to know about a particular literature related to your gene, you can utilize these databases to know about the literature. And the last thing is data integration analysis. So of course, if you want to know about the sequences related to your query sequence, you can utilize these databases and you can integrate a particular information in a database, and in return you can get a particular information. And if we talk about, it's very common among the students, if we talk about the NCBI BLAST, there you actually insert a query sequence, and in return you specify a particular database, and in return you get information about the alignment, you get information about the template that are associated with your sequence. So bioinformatics databases also help in the data integration analysis. Now, what are the challenges and limitations that are associated with bioinformatics databases? The first thing is the data quality. Of course, the influx of the data is in a very rapid way, because experiments are going on. So there is a point that the data quality is compromising. Like just to give you an example, sometime if you want to get an information about breast cancer, you can utilize the Kaggle database, but you cannot get enough information about that particular database. There will be information like just to give you an example, if you want the information about how the how insulin is related to the breast cancer, or how insulin is related to the progression of the breast cancer, you cannot get enough information, because the quality the quality of the information is not enough to give you a symmetrical or coherent information. The next thing is the data integration. Sometime you want to get an information about sometime you want to actually blast a particular sequence against a particular database, but that database is not available. So yeah, there is some challenges that are associated with the data integration. The second last point is the data interpretation, and I think one of the biggest challenge when it comes to the bioinformatics databases is the data interpretation. There are a lot of the databases, but for the layman, it is very difficult to understand, and it's very difficult to interpret. And there is a famous saying that 20% of the work is done on the part of the process, and 80% of work is done on the part of the interpretation. So data interpretation is very challenging task when it comes to the understanding the bioinformatics databases. And yeah, the last point is the technical interpretation. There are databases like when we talk about the Bootcamp script, it is the script that is written in our language, but for the biologist or for the biology student who is not good at the technical stuff, who is not good at utilizing the technical stuff, it is very difficult for him to understand the Bootcamp package and to run that Bootcamp package for the gene expression analysis. So these are some of the challenges and limitations that are associated with the bioinformatics databases. Now the last part is the future direction in bioinformatics databases. So we talk about the challenges. So the first future direction is maybe in future there will be an integration of multi-omics data. There will be a database where you can get information about the protein sequence, and at the same time you can get information about the pathway that is associated with that particular sequence. So there will be integration of multi-omics data. The second part is the development of machine learning algorithms. Yeah, so there is going to be a database where you can get information by one click away. Like if we talk about recently, there is a paper published about the GPT. So soon there will be a database that will give you information with one click away. There you don't need to navigate to multiple tapes to retrieve a particular information. And the third point here is the development of the personalized databases. A personalized databases, what I mean by personalized databases is there will be a database just for the cancer, there will be a database just for the Homo sapiens, there will be a database just for the particular disease. So in future there will be some development around these personalized databases. And the second last point is the use of the blockchain technology. Of course, if you talk about the NCBI databases, NCBI is some sort of a regulatory authority that actually regulate the data that comes from the hospitals, and it connects the data that comes from the hospital, and it gives you the data to the scientific community. So maybe there will be a system to directly connect the hospitals to the scientific community. So the use of blockchain technology will soon going to happen when it comes to the bioinformatics databases. And yeah, the last part is the continued focus on the data sharing and open science. There are databases that actually need a subscription to access the data. So there will be a continued focus on the data sharing and open science part. Now, I think that's enough about the bioinformatics databases, and this particular slide explain you do not need to worry about the concepts. You do not need to worry about how you are going to learn these databases. It's all about exploring the database. Yeah. So these slides actually explain what is actually a bioinformatics databases and how they are important and what are the types of bioinformatics databases. Now let's explore some of the databases. Here you can see I've already opened the protein data bank, and when we talk about the core features of the bioinformatics databases, there we mentioned there will be an option for the search, there will be an option for there will be a lot of the options to access the data. So here you can see I have a search option, and I want to search let's say I want to search about the proteins that are associated with this particular species, Aspergillus, which is actually a fungi. So let's search the protein data bank, and it will take some time. Now here you can see we have a lot of the options here related to the Aspergillus species, which is actually a fungus. So let me give you a little information about this one BK1 is actually accession number or identification number that is associated with this particular protein. Now let's open this this protein. And here you can see we have information about the PDB DOI, which is actually again it's actually the address that is associated with this protein. We have information about the classification, organism, the expression system, mutations. We have also information about the experimental work, like the method that is used to build this particular 3D model is the X-ray diffraction. Resolution is 2 angstrom. These informations are very important when you are working on more than one protein. You have to know about the method, you have to know about the resolution, you have to know about the organism, and you have to know about the classification as well. Now this part is about the literature. If you want to know about the literature part associated with this particular protein, you can click on this particular link and you can read the information accordingly. Now here you can see we have information about the chains. There are chain 1, the sequence length is 184, the organism is Aspergillus, and other details like the image also. Here you can see this is a very important information. We have the reference sequence. This is the feature view of this particular protein, and the most important thing here for you to notice is the unmodeled region. So what does it mean by unmodeled residue? Unmodeled residues are those residues that are not modeled. The sequence is there, but there is no 3D structure. So these unmodeled residues, they are very important when it comes to the 3D structure prediction. So this is about, you can zoom in, you can zoom out, you can get information about the disorders, you can get information about the disordered binding. So this is about the PDB. Now let's talk about the NCBI. This is the biggest and the most famous database. This is actually the National Library of Medicine, National Center for Biotechnology Information, and this database is this is a primary database. You have secondary databases like assembly, biocollection, bioproject, biosample, books. You have conserved domains, the DBWAR, the gene, genomes, geodatasets, geoprofiles. So these are some of the secondary databases that are associated with the NCBI. You can select a secondary database and you can type your query here in the search box accordingly. Now let's talk about this particular database, the KIC. If you want to know about the pathways that are associated with a particular gene, you can utilize this KIC database. So that's it for today. Thank you for watching this tutorial, and have a great day. Thank you.